reproduction and life cycle of Nitella species. Nitella is a Carassian algae and it has got, just like other Carassian algae, it has got a equisetoid habit and is oogamous in reproduction. Hello friends, I am Dr. Ayantika Das and in this video I am going to discuss about this Carassian algae Nitella species reproduction and life cycle. I have made an introductory video earlier of Nitella species where I have discussed the overview, distribution, morphology, anatomy, the economic and ecological importance of Nitella species, its fossil records, as well as its control measure. So here I will be only discussing about reproduction and life cycle. I have put the link of the earlier video in the description box and you can go and check that video also for a better understanding of this video. So without any delay, let's start this video. Now the reproduction, we know that all the Carassian algae, they have a characteristic that they do not have any asexual reproduction. That is, there will be no zoospore or aplanospore. But however, they do have this vegetative reproduction. Vegetative reproduction can be either by fragmentation, which is very common for all algae, where any fragment, any portion of the plant that is algae which is getting fragmented by either the wave action or by the gnawing action of herbivorous aquatic invertebrates as well as vertebrates. So these small fragment, whether it is the nodal part or the internodal cell, doesn't matter, any part, nodal, internodal part of this Nitella can give rise to entire plant. Then after that, after this fragmentation, we see secondary protonema in vegetative reproduction. Now we know that primary protonema are formed when the oospore of Nitella germinate. But this secondary protonema, they are formed when any perinating body of Nitella, it can be a root portion, it can be a rhizoid portion, it can be a shoot portion or rhizoid portion. Any portion can, after a period of dormancy or perination during the hot dry season, when return of favorable condition occur, this old plant, any portion, either shoot or the rhizoid portion, they give rise to protonema like structure, which is here. This protonema like structure is formed, and from this protonema the new cells or a new vegetative body of Nitella is produced. So that is the secondary protonema here in this picture. How the protonema looks, it is tube like and much branched. So this is the secondary protonema, and this is also a very important process because in countries, tropical countries like India and the Southeast Asia and South Asia, we are seeing that during the summer month, both Cara and Nitella, they disappear. And after the rain or during the rainy season, this type of secondary protonema are observed. So these protonema, they arise from the perinating body, any portion, doesn't matter, it will give rise to a secondary protonema. Then we have tuber. We know that while studying Cara, of which I have also made a video in two parts, one is the introductory part and another is a reproduction. So link to that also I have put in the description box. So just like Cara has got amylum stars, tubers and all. So in Nitella, we are seeing only the tubers, and these tubers, they arise from the rhizoidal portion, not the basal nodes, but just the rhizoidal portion. The tubers, they are starch-filled structures, and they are they form an outgrowth-like structure on the surface of the rhizoids. and these are called the tuber like structure. For example, let me draw. If this is the rhizoid of Chara is much branched multicellular. So if this is the rhizoid, tubers will be, this tuberous outgrowth will be present in the surface of the rhizoids. So these are basically the tubers. So this is about the three vegetative reproductive modes. Now we come to the sexual reproduction. In all the Caracean algae, that is the Chara, Nitella, Tolipula, any Caracean algae, there is one characteristic feature, that is reproduction will be oogamous. This is characteristic of this class. And as the reproduction is oogamous, they will have antheridia and oogonia. Here the antheridia is called nucule. It is, the antheridia is called globule, which is globular in structure, and the female organ is called nucule. Now this Chara as well as Nitella, they have got a characteristic, that is some of the species they are homothallic, that is the single plant of Nitella will bear both the sex organ, or they can be heterothallic, that is a single plant will bear only a particular type of sex organ. So that is they are unisexual. So both homo as well as heterothallic form of Nitella found, and these are the examples of the homo and heterothallic species in Nitella. So we see that the sex organ, the position of the sex organ, they are born on the furcation of the on the furcation on the leaflets or branchlets, we can say on the leaf or branchlets. So if this is the main stem, so this is the main stem of Nitella, and this is a branchlet, and we know that branchlet has got furcations. So we see that in this furcation area, among the furcation, the sex organs are born. So these all black colored body, these are all sex organ that is born on the furcation. Here there is a detailed example, a diagram, where we see that these are the nucules, and globule is born on the top, on the terminal position. For example, this is the globule. If it is born, if it is a bisexual or homothallic species, then it will contain nucule. These are the nucules. These are the female reproductive organ, that is the nucule, and this is the globule, that is the male reproductive organ that is born among the furcations. So if this is the furcations, they are sex organs are born like this. So we see there is a terminal nucule. I am drawing it in a different color so that you better understand. So if this is the globule and these are the nucules, that is the globule is terminal in position, and nucules, they are lateral in position. So let's first talk about the globule or the antheridium. So the terminal apical cell, they serve as an antheridial initial and divide twice to form an antheridial mother cell and the pedicell, which undergoes several divisions to form this antheridial mother cell undergoes several division to form this jacket layer as well as inner antherogenous cells, and later the final structure, it looks something like this. Better to understand it here. So outwardly, the outward appearance, external appearance of the globule is like this. And if we take a transverse section, we see that there is an external jacket layer which is called a shield cell. And this there are two type of in Nitella there are two type of species, where some species has got 8 number of shield cells, some species has got 4 number of shield cells. So these shield cell can be either 4 or it can be 8, depending upon the species. It can be either 4 or 8. If the Nitella has got 4 shield cells, then it is called a quadriscutate variety species. Or if it has got 8 cells, then it will be called octoscutate. Most of the Nitella species are octoscutate. That is, they will contain 8 number of shield cells. Now, after the shield cell, we get the manubrium. The next layer is the manubrium. And if this is the shield cell layer, we can see that we have told that either 8 or 4, but we are seeing a large number of cells. These, but these are not a separate cell. Actually, the shield cell, it has got the cell wall of the shield cell, as well as a plasma membrane. It has got invagination, giving an appearance that there are many a number of cells, but these are not separate cells. These are a single cell of shield cell. A single shield cell, by invagination of the cell wall, they form this structure that gives a false illusion, that they gives an illusion of many celled, but this is only a single cell. So after that, we have, let me take another color, we have the manubrium that I should be drawing in, say, red. So this, after the layer of shield cell, we have this manubrium, which is also very common in Cara. Also, we have seen this type of manubrium. In Cara, this manubrium was club shaped, but here it is a bit tabby. It is not that much elongated. So this is the manubrium. Here we see this is the manubrium, this cell. So after the manubrium, there we see that only there are 4 manubrium, 1, 2, 3 and 4 manubrium cells. So after that, we have got the primary capitulum. Primary capitulum is born next to the manubrium. Let me draw it in another color. So these are the primary capitulum cells that I am drawing in green. Then after the primary capitulum, each manubrium has got a single capitulum. So there will be 4 primary capitulum, and next to the primary capitulum will be the secondary capitulum. Each of these 4 primary capitulum will bear again, each of this will have the 6 number of secondary capitulum. So there will be another 6 number of secondary capitulum that I am drawing in yellow. So this will be the secondary capitulum cells, and the earlier that I have drawn in green, it is the primary capitulum that I am writing as PC. So we see that the secondary capitulum has each of this primary capitulum give rise to secondary capitulum cells, which in turn bears the antheridial, the antheridial filament that I am drawing in blue. So these are the basically this structure here is the antheridial filament. So each of these antheridial filament, so if this is the antheridial filament, they bears a series of cells that give rise to the sperm. From each of these cells arise the spirally, from each of this cell, the spirally arranged sperm is present, and this sperm, each of the cell, when the sperm is mature, then the sperm, either the wall of this antheridial cell, they gelatinize, and there is a hole through which um, the sperm comes out, or the sperm, it dashes against the wall and create the hole through which it escapes. So there is two process by which the sperm escapes early in the morning from the antheridium. Now this is the structure of the sperm. This is a characteristic of structure of all Carassian algae, and that is this spirally coiled sperm body, much elongated, tubular, spirally coiled. And this type of coiled body, they have got a flagella for movement, two flagella. We see that both these flagella here, these two flagella, they are of similar structure, but there is a slight difference in their size. One is much smaller and another is larger. And these flagella, both these flagella, they have entire body of the flagella is covered by this diamond shaped scales. So this entire flagellary body, they are covered with scales, and the two flagella of unequal length, they are placed in the anterior portion, that is this portion, they are anteriorly placed, but their direction, they are posteriorly directed. They are anteriorly placed but posteriorly directed. And so entire sperm body is divided into the head or the rostrum portion, that bears mostly the cytoplasm that is rich in microtubules, and bears the two flagella. So this is the head or rostrum portion. Then the next portion is the middle portion, that contains this. If this is the middle portion, so we see that middle portion has got nucleus, mainly elongated nucleus, and with nucleoid, this one. And the middle portion has got a very thin layer of a dense cytoplasm that is filled with microtubules as a embedding the nucleus, and this thin layer of cytoplasm lies between the membrane and the nucleus. So, and this cytoplasm has got many microtubules. So the middle portion, they have got only nucleus and microtubule rich cytoplasm. And the nucleus also, it takes 2.5 to 3 turn, as the number of turns is present in the sperm. And then we have the posterior portion, and the posterior portion is also coiled and covered by this scale like structure. These are the scales, diamond shaped scales that cover the entire body of the sperm. And in this posterior portion, other type of organelles are present. For example, mitochondria, they have vesicles, they have plastids, and this we know that the plastids of all the Charophycean algae, they do not have pyrenoid. So the plastids do not contain any pyrenoid, but they the starch filled area, they are in the vesicle. The starch is stored in the vesicles. This starch is stored in the vesicles. I've written a wrong thing. So there is plastid, but the starch is filled in the vesicles, and they also have a rudimentary enzyme pellet in the posterior portion. So this is the enzyme pellet, this portion. So this is how the sperm structure looks. And this is the nucule. that is the female reproductive organ, the oogonium, where we see that the oogonium, they born at the nodes, and just like the globule, nucule is also born at the nodes, and it has got, we see, these are the spirally arranged tube cells. The tube cells, there is 5 spirally arranged tube cell. So this is the tube cell, and at the top of it, let me draw it in another color so that you understand better. So at the top there is this 2 hole of corona cells. In the Cara nucule, they have got only 1 hole of 5 cells of corona cells, but here we will have another hole, that is 2 holes of 5 cells. So it will be looking like just like a flower. So there will be 2 layer in 2 holes. There will be 1 hole of corona cells, and again there will be another hole of corona cells. And these 12 corona cells, these are the corona cells, and that is arranged in 2 holes, and these are the spirally arranged, right-handed spirally arranged. These are the tube cells that are spirally arranged. They are spirally arranged like this. So these are the red. In red I have drawn the tube cell, and in yellow the corona cell. Inside to it we see another egg cell. Inside to it we see, this is the egg cell. This one is the egg cell. And this egg cell has got a very characteristic receptive spot. This is the egg cell, and it has got a hyaline receptive spot here in the anterior region, and in the posterior region it has got nucleus. So antheridial initial, the nucule, the oogonial initial is any lateral cell. Earlier we have seen that the terminal cell is the antheridial initial, and the lateral cells, these lateral cells, these are the oogonial initial. So this is the oogonial initial. So this oogonial initial divides, and there is by division the pedicel, which takes place no further division in the pedicel cell, and the oogonial initial the oogonial initial, they again divide and redivide, and the terminal cell, this one, they ultimately give rise to this cell. This terminal cell give rise to the oogonium, and the immediate next cell, they undergoes division to form the tube cells and corona cells. So this sheath cell, they give rise to this one. They divide and redivide to form the tube and the corona cell. So this is how they are formed, and this is always this cell give rise to this one. So there is a receptive spot through which this sperm fuses with the egg, and this otherwise this receptive spot is quite less granular and hyaline. Otherwise the entire, so the fertilization, when the nucule and globule all they are mature, so in the nucule, that is the female reproductive organ, the tube cells that are spirally arranged, I told you, these tube cells, they separate from each other. So these tube cells, they separate from each other. So some splits will be created in this area. So the sperm that is released early in the morning, they swim through these slits. The sperm, they swim through these slits, and they swim and they enter. Let me draw the sperm in another color, yellow. So the sperm, they come swimming and they enter this through the slits, and then they swim to the top portion here, and in the hyaline area, this area, they penetrate the egg in the hyaline area. Later it sinks down where the nucleus is present, female nucleus is present, and fertilization occur. So after the fertilization, say for example, this is a fertilized oospore, that is a fertilized zygote. So after that, entire outside of the egg, there is a membranous deposition, and this deposition is cellulosic in nature, and this, at this stage, it is called the oospore. It is called the this is the oospore. So this is the oospore. So after fertilization, when the membrane is laid around the egg cell, the zygote, then this cellulose containing membranous structure, which is diploid, is the oospore. And apart from that, oospore is also has an investment of these tube cells, and on the top of these tube cells, on the outer surface of these tube cells, deposition of calcium also gets deposited. So after fertilization and the formation of oospore, this stalk cell, they separate from the rest of the plant, and it fall to the bottom of the water body. So it gradually sinks to the bottom of the water body in the sediment and lies there in dormancy if there is not much conductible condition, and if there is good amount of moisture still, then also there will be a period of rest, and after that period of rest, further germination occur. Now, during this period of rest, the oospore, they change color from yellow, golden yellow, to gradually brownish to ultimately black in color. So after the period of rest, the oospore gets activated. If there is water around it, it gets activated, and the oospore, the single nucleus of oospore, they undergo meiosis. This is meiosis. Meiosis, that is the reduction division. Reduction division, where this was 2n, but this was 2n, the oospore was 2n, but after the period of rest, when it germinates, there is reduction division of meiosis, and this contain haploid nucleus. That is, after meiosis, all these 4 nucleus that is formed, they are all haploid. And of these 4 nucleus, again there is a division, the upper cell, that give rise to the protonema, and the lower cell containing these 3 nuclei. The nuclei later degenerate. These are the degenerating nuclei, and the upper cell containing a single nucleus undergoes an oblique septation. The wall is laid down obliquely. The upper cell, this one, the upper cell, they form the primodia. The upper cell form the primodia for the protonema, and the lower cell form the. So this germination we are seeing, this is a very good picture. That is, the primary protonema is moving in the upward direction, the rhizoidal in the downward direction, like this here, and this is the bipolar germination, And this is how pretty much this primary protonema later forms nodes and internodes here, and it gives rise to the entire Nitella plant. So in the entire life cycle of Nitella, here I have taken the diagram from Cara, so there should be another correction. This is the globule portion of Cara, but in Nitella this will not be the exact position. The exact position of the globule in Nitella will be terminal. This is the globule of the Nitella and this is the nucule. So in Nitella, the difference between Nitella and Cara is that in Nitella you have terminally placed male reproductive globule and laterally placed female reproductive nucule. So when this sex organ they mature, the sperms they are released and that entered in between the open region that is formed by the tube cells. They separate from one another, creating slits. So the sperm they swim and through the tube cell, opening in the tube cell, and then they swim to the top region, that is the receptive spot region, and then they gain penetration through this receptive spot. They fertilize and this is the zygote and this is the storage cell or oospore. So this is the storage cell or oospore. So both the zygote, we know that it will be diploid after zygote that is formed after fertilization is diploid, and the zygote when undergoes a period of rest, then it germinates. So during the period of rest, this storage cell is called the oospore, which has got a wall around it, the cellulosic wall around it, and lime encrustation on the surface of the nucule. So and it lies at the bottom of the lake sediments and rests. So the entire period, then after the period of rest, it undergoes meiosis, and then this is this germinating protonemal stage. This becomes again haploid. So entire life cycle of Nitella, after fertilization, the zygote and the oospore, these are the only diploid portion in the life cycle of the Nitella. So after that, after meiosis, this germinating protonema and the rhizoid, they ultimately are forming the entire Nitella plant. So this is how the life cycle occurs, and this is about the sexual. But if we see a vegetative reproduction, then we see there are three types of vegetative reproduction. One is the fragmentation. There is again secondary protonema and tuber. Pardon my bad handwriting. And then you have this secondary protonema, that I am writing just secondary protonema as secondary P. So these are the three vegetative reproduction, and this is the. So this was all about reproduction and life cycle of Nitella species, which is a Charophycean algae. I've made video on other type of Charophycean algae also, link I have put in the description box. And I have also made many other videos on many algae, and you can go to my channel and watch those videos. I've also made videos on fungi, on the bryophytes, pteridophytes, on ecology, on lichens, on cyanobacteria, bacteria, you can go to my channel and have this videos in the it has been put under different segments, and you can just go and watch those videos. If you like this video, do share it with your friends, and do subscribe to my channel to get more such videos. And also I'll be very much encouraged by your subscriptions. Those who have already subscribed, thanks for subscribing, and thanks for watching this video.